I would now like to invite Professor Nadira Karunavira to deliver the presidential address for the year 2021.
Distinguished invitees, past presidents, board of directors, fellow council members, and members of the Sri Lanka College of Microbiologists, including those joining us online, colleagues, friends, ladies and gentlemen. First, let me thank Shirani for that generous introduction, and let me find the pointer first, and then share with you the theme for the scientific activities of the college for this year. It is the threat of new and re-emerging infections, role of novel tools and technologies to face challenges. So, to be in line with this theme, what I'm going to talk to you about is on this topic, man versus parasites, weapons to combat. There are zillions of organisms that continue to affect human health since time immemorial. Majority are harmless. However, there are disease-causing or pathogenic organisms which could be broadly divided into viruses, bacteria, and parasites. Infectious diseases, I'm sure you will agree, cause a very heavy toll on human health. And these figures given here are actually pre-COVID figures. I'm sure it must have tripled or drastically increased by now. So the human-pathogen relationship is often recognized as a co-evolutionary arm race, each trying to outsmart the other. So in this backdrop, I would like to take two parasitic diseases as examples and explain to you a few studies that we have done in the field of malaria and in part 2 in the field of leishmaniasis. The studies, selected ones, aimed at developing and testing novel tools for efficient disease management and control. In other words, our attempts at expanding this arsenal that we could fight with. To start off with malaria, it's a mosquito-borne disease, and Sri Lanka has been quite successful in shrinking the malaria map. In fact, by end of 2012, Sri Lanka was able to get rid of all locally acquired cases of malaria. And in view of this success, the World Health Organization awarded the Certificate of Malaria Elimination to the country in 2016. And it was a huge achievement, especially when one considered where we were in mid-1930s, before I think most of us were born. There have been millions of cases of malaria with nearly 80,000 deaths at that time. We can't even imagine that scale from where we stand now. So one might wonder why we need malaria studies if we have achieved elimination. If you look at this figure, which gives the number of malaria cases, the dark blue bars indicate the locally acquired ones, and there were no cases after 2012, as I said, after achieving elimination. However, these light blue ones, which indicates imported malaria or traveler's disease, continues. And these patients come, or people who are carrying this traveler's malaria, they come from various countries, but majority are from the region, India, Pakistan, and so on. And if we look at the numbers that are brought in, around 50 cases are encountered each year. So what are the risks involved in malaria being brought in by travelers? It is because of the persistence of vector mosquitoes in the country. Although we gotten rid of the cases or locally acquired cases, the mosquitoes still persist. And there are susceptible hosts, that's you and I, who remain susceptible to acquire malaria. And also suitable environment for malaria transmission, particularly in the dry zone of the country. Therefore, introduction of malaria cases through travelers poses significant risk of re-emergence and resurgence of this disease. So, quite commendably, the services of the anti-malaria campaign, Ministry of Health, has continued in the post-elimination period with a particular focus on active surveillance to detect and treat cases, to look at vector surveillance, vector control, and so on. And we also have continued our studies both during the pre- and post-elimination era with a focus on developing and testing new tools to facilitate disease management and control. So I have selected a few studies, couple of studies that we did on genotyping of parasites in order to track them to their geographic origin, which would be a tool that one could use to strengthen surveillance in the post-elimination era in Sri Lanka. These are the two species, Plasmodium vivax and falciparum, which are the two common malaria parasite species that cause malaria in the world. And we focus on Plasmodium vivax because it is the more prevalent species in the region, in Asia. And what we did was we examined the genome of the Plasmodium vivax parasite and selected genotyping markers in the form of microsatellites, which are nucleotide repeat regions in the genome, which could be used for genotyping. And the conditions were optimized, and these are actually the PCR conditions that were used to amplify these loci. And this table just gives the summary of these details on the loci used or the markers used, indicating where they are located in the genome, the nucleotide repeat regions in each one of them, and the primers used. So what we did was we tagged these or labeled these primers with fluorescent dyes so that the length variation of the labeled PCR products could be visualized using capillary gel electrophoresis using this machine called the ABI sequencer. So the reading that we got, the output of this machine provided this sort of picture, which we could use to read off the size, length variation or the size of the PCR product using this scale. So it was something like this. So it was possible to look at length variation of these alleles or the haplotypes that we looked at in different isolates that we tested. So we tested isolates from Sri Lanka. This gives the number of isolates tested. Ethiopia and Myanmar, again, where Plasmodium vivax is endemic. So we got a whole heap of results, which was very informative, given the number of alleles, diversity figures, and so on, which I am not going to dwell into. But I will try to explain the population structure studies that came out of it. And we did the analysis using structure software to look at the clustering of these haplotypes or the alleles according to geographic origin. So what we got was something like this. This figure actually summarizes the results, which indicated basically that if you look carefully, I'm sure even at a glance you can see how these parasites that come from these different geographic regions vary from each other. So there was clustering of these parasites according to geography, which was very interesting to see. And this work was primarily done by Professor Shamini Gunawardena as part of her post-MD training. So as I mentioned, there were loads of information regarding the polymorphism, the diversity, clonality, and so on. But I would like you to focus on the last point, which simply indicated that microsatellite typing is indeed a useful tool that could be used for predicting the geographic origin of an isolate, and also it could be used to map outbreaks of malaria, which remains as a challenge even in countries that have wiped out the disease. We didn't want to stop there because microsatellite typing isn't an easy method to perform. It's a laborious technique. So we were on the lookout for more efficient tools. So we found one in the form of SNPs, or single nucleotide polymorphisms, that could be used to identify genetic variations within species using very short genetic sequence from standard part of the genome that contain that SNP. And we use this method called SNP barcoding, which contains a combination of selected SNPs that together express a unique pattern of variation. And the concept is basically what one would use to identify products in supermarkets. I'm sure this might look familiar to you. So basically what they do is they scan this at the supermarket counter to get the details of the product, including the price. So the concept used in SNP barcoding was something similar to identify these different alleles. So it was possible to compare the genetic diversity between populations using software called principal component analysis, and it enabled us to identify these alleles or different isolates that varied together across samples, something like this. So when we got the barcoding done on all isolates that we tested, and we arranged them according to the similarity in genetic composition, we got a picture like this, which was a bit complicated. And in order to simplify, we used principal component analysis again that generated figures like this that enabled us to demonstrate that these alleles can be separated out according to geography. Simply, it was possible to conclude that SNP barcoding can be used to distinguish locally acquired or indigenous parasites from imported ones, and therefore will be useful to track imported malaria cases. And this work was done primarily by Rajika as part of her PhD. She completed her work recently. I gave you only a snapshot of these studies because of the time constraints. If anyone is interested in details, one could find in these publications. To move on to the next part on leishmaniasis, which remains as a relatively new health challenge in Sri Lanka, we were interested in looking at new tools to test them, validate them, to facilitate better patient management and infection control. We were interested in looking at the causative agent, which is the Leishmania parasite, in order to understand this new disease, and also looked at ways to dissect out the epidemiology, disease burden, and pattern of spread to help plan future control measures, and also find ways to enable better patient care or patient management. So before I go into details of each of those areas, let me give you a background to this disease. Leishmaniasis is not a single disease entity. It is a range of diseases with varying clinical forms, and it ranges from mild form, which is confined to the skin, to more severe fatal forms that affect organs, which is called visceral leishmaniasis. And this is caused by the Leishmania parasite and transmitted through a tiny insect called the sandfly. And it's a disease that is found in many tropics and subtropics. In Sri Lanka, the first case of skin form of disease or cutaneous leishmaniasis was reported in early 1990s. However, things were fairly quiet until an outbreak was reported in 2001 from Valioya, that is in the north central province. And subsequent studies indicated that the causative agent is Leishmania donovani, which is the same species that is found in regional countries, which cause a very different form of disease, more dangerous form of disease in those countries. And the vector is the sandfly, Phlebotomus argentipes, and this shows much enlarged view of the sandfly. It's a tiny insect. In real life, it is the size of about 1/3 of a mosquito. So you can imagine how tiny it is. But if you look carefully, I'm sure you might identify these insects from your backyard because they frequent these areas with vegetations, and they are found almost all over now. And the parasite is a protozoan parasite. It is a single-celled organism that is found inside host mononuclear cells, and these are mononuclear cells that you see here. The nucleus is here of the cell, and these tiny blue dots are the parasites. So this is again a hugely magnified view. Quite thankfully, predominant clinical form that we see in Sri Lanka remains as the skin form of cutaneous leishmaniasis. And these skin lesions are characteristically non-itchy, they don't itch, and also they don't hurt, not painful. However, they are found on exposed areas of the skin where it's accessible to sandfly bites. So you get the lesion at the site of the sandfly bite. And these are the types of lesions that we see frequently: tiny papules, nodules, or ulcers. And how are they treated? The most frequent drug or commonest drug used is sodium stibogluconate, which is given as intralesional injections. In other words, it's injected into skin tissue surrounding the lesion, and it's a fairly painful procedure. You need weekly injections for several weeks, for about 3 months in most cases. And to make matters worse, there are more and more cases that are reported with failure to respond. And it is alarming when a recent report giving these drug failures of over 75% of cases. So it's not a very encouraging picture there. Cryotherapy was used before SSG or sodium stibogluconate was brought in. Cryotherapy is simply use of liquid nitrogen that is sprayed onto the lesion. Again, you have to repeat it at bi-weekly intervals for several weeks, so it's not a very pleasant form of treatment either. There are other drugs like amphotericin B that are used. Again, those are toxic and not very popular. In addition to the skin forms, we also have seen other forms of disease in Sri Lanka, like the ones that affect the mucosal tissue. Here it's the upper lip. And in this patient, it was rather serious because he also had tuberculosis and other complications related to immune deficiency. And leishmaniasis has caused this total destruction of the nasal septum and also the upper palate, which was a really complicated case. There have been few cases of visceral leishmaniasis reported from Sri Lanka as well. And as mentioned earlier, it's a serious disease which affects the organs like liver, spleen. So unless you detect these patients very early during the infection and treat them effectively, they succumb to this disease. So it's a potentially fatal disease. The causative organism is Leishmania donovani, and it is better known to cause visceral disease in other countries, including India. In fact, ours was the first isolation of this particular strain from skin form of disease. So we were interested in dissecting it out using molecular methods. This was, we did molecular typing using microsatellite markers again. And it was, this is a phylogenetic tree that shows the relationship, genetic relationship between parasites from different regions. And as you can see, the Sri Lankan parasites are quite close. The genetic structure is quite close to those in the neighboring countries like India, Nepal, Bangladesh. However, they have separated out. They have a distinct genetic structure. And this might at least partly explain why we are seeing this different clinical form or phenotype. And this is the work of Dr. Yamuna Siriwardana, who did this as part of her PhD back then. And this work was continued to look at the sequence diversity or sequence differences in different clinical forms that we are seeing in Sri Lanka. These are the isolates that were derived from cutaneous leishmaniasis patients, and these were from visceral leishmaniasis isolates, and these from cutaneous leishmaniasis that did not respond to the regular drug SSG. And as you can see, the sequence analysis, again, this is a phylogenetic tree. It demonstrated that they could be separated out. So the genetic structure appears to be different in each of these phenotypes. And this work was done by Udeshika, who completed her PhD recently, and now she's a postdoc at Johns Hopkins University in the US. Now this, we went on to sequence, that is to obtain whole genome sequencing of selected isolates from Sri Lanka. And this is actually a heat map that shows the grouping of these isolates that were sequenced based on their SNP profiles. The sequencing was also done in the local genetics lab in our faculty, which is headed by the current dean, Professor Vajra Dissanayake. And this shows the SNP profiles, analysis based on the SNP profiles. And if you look at the associated dendrogram, you can see like three different clades or three different groupings. Again, these were due to the different clinical forms, the cutaneous, visceral, and also the query drug resistant or poor responding patients. And this work was done by Sumudu and Dr. Nilakshi Samannayaka, who is a senior lecturer in our department. So in this backdrop, you will be interested to hear that there's an ongoing program of elimination. It's called Elimination of Leishmaniasis from the Indian subcontinent. And it's a program facilitated by the World Health Organization, which was started in 2005 with governments of India, Nepal, and Bangladesh signing up for this program as partners. And there were a couple of other countries that joined in this program later on. However, Sri Lanka is not part of it yet. And if you look carefully, this gives the number of cases in each of these three countries that started this elimination program. And from 2005, when they joined the elimination program, the picture is quite encouraging. There's a downward trend, that is reduction in number of cases. Although they still haven't reached elimination targets, the countries remain hopeful that they will do so within the next few years. In contrast to this picture that is seen in the region, in Sri Lanka, the case numbers have continuously gone up, particularly during the last few years. From this 2018 onwards, the 2020 figures are somewhere here. So it's not very encouraging. And we also looked at we used mapping tools to look at the disease burden and spatial spread of this disease from 2001, that is beginning of this outbreak onwards. So this is 2001 and it goes this way up to 2018. And if you look even at a glance, now use this color coding to detect the burden, which says the intensity of burden in each of these districts. As you can see, the outbreak started here in the north central province, and it has spread thereafter. And if you look at the color coding, you can see even at a glance how the colors have become darker, pink appearing, then the red, and then maroon, and so on. So, and there's spatial spread as well, affecting all the districts in the country eventually. So, when we put all that data together, you can see that all the districts have had cases. However, most number of cases are from these 3 districts in the north central area and Matara and Hambantota from the south. In fact, these 5 districts accounts for over 85% of cases that are reported from the country. And this shows the trend of this disease in each of these districts, which closely mimic the trend that is seen in the entire country. We used hotspot analysis to look at these hotspot areas or high burden areas in the north central and southern part. And these figures are from 2015 onwards. And as you can see, in the north central area, the hotspot has shifted and it also has shrunk. But in the south, it has expanded and the disease burden has become even more as years pass by. In addition to looking at the parasite and the epidemiology, we also looked at new tools for patient management in the form of thermotherapy. We did a controlled clinical trial using this machine called the thermotherapy machine, or it is also called radiofrequency heat therapy. This emits radiofrequency waves which comes through this wire, and through the electrode it is introduced onto the skin surrounding the lesion. And the principle behind it is these radiofrequency waves cause rapid molecular movement in the skin tissue, which generates heat, which stimulates a good immune response and induces cure of the lesion. So that is the principle behind its use. So in the clinical trial, we used 2 arms. I know you can read this. Don't even try. But this is just to show that in the control arm, we use the regular drug, intralesional sodium stibogluconate, weekly injections up to a period of 3 months. And in the test arm, we use thermotherapy or radiofrequency heat therapy, which was applied only once at the beginning of the trial. And these patients were followed up weekly up to a period of 3 months, and less frequently thereafter up to 1 year. Just to summarize the findings, so the comparison was between single application of thermotherapy versus weekly injections of IL-SSG injections. This thermotherapy arm, the lesions healed rather fast. However, as time passes by, the healing rates became similar. So by end of 3 months, it was comparable. So in the key findings of this trial were thermotherapy is an effective treatment method for cutaneous leishmaniasis in Sri Lanka, and a single application was found to be safe and effective, and probably importantly for a country like ours, it was 7 to 8 times cheaper, the cost per patient, and it is patient friendly as well because they need to come to the hospital only once to get the treatment, as opposed to multiple doses of intralesional injections. So we replicated this trial on poor responding patients to SSG because it's a huge problem in the field. and that trial, the second trial, demonstrated similar efficacy and safety in those patients who have not responded to sodium stibugluconate. The first trial was done by Dr. Varda Refai, who is a parasitologist at MRI. She did this as part of her MD project. And the second one was done by Dr. Hermali Silva, who is a lecturer in our department. So in leishmaniasis, there's a notable increase in the case burden and spread since 2001, with a prominent increase in 2018. And there seems to be a steady expansion in risk areas with persistent 2 hotspots in the north central and southern parts of the country. Thermotherapy is a cost-effective and patient-friendly alternative that we could use to overcome challenges that are faced by clinicians in patient management, including those who fail to respond to SSG. And there's evidence of subspecies diversity in this parasite, and we are pursuing this work to look at this diversity, how this diversity translates into the different clinical forms of pathogenesis and implications on patient management and treatment outcome. So finally, to get back to the title that I started off with, Man versus Parasites, Weapons to Combat, our attempts at expanding the arsenal that we can fight with. In the case of malaria, it was possible to show that genetic markers, particularly SNP barcoding, is a useful tool for genomic surveillance to track parasites to their geographic origin, which could easily supplement the current patient surveillance that is done based on patient travel history alone. In the case of leishmaniasis, genetic tools are available for parasite typing and at subspecies level, which could be linked to disease pathogenesis. Spatial and epidemiological tools are ready to track and monitor hotspots of disease, which can assist and even direct any future control programs that can be put in place. And novel treatment options are available as safe and cost-effective treatment method for these patients. So finally, to come to recommendations, in the case of malaria, in the post-elimination period as we are in now, one could introduce more efficient methods for parasite surveillance to aid management of travelers' malaria, which remains as a challenge to the country. And to contain the spread of leishmaniasis, it is required to enhance the surveillance, public awareness, and interventions are urgently needed in targeted areas to halt this outbreak of disease. And also, review of existing treatment protocols are required with new treatment options to be considered. Before I end, I would like to thank all those people who are responsible for my existence. To start off with my parents, my late father, who is shown here, my mother, who is in the audience, my husband, Cecil, 2 daughters, Aisha and Sajani, my brother Nalin and sisters Purnima and Sadhana. I'm so thankful for their love, understanding. I'm particularly grateful to Cecil and the 2 girls for their enormous patience and putting up with my crazy schedules. And in my career, whatever the distance that I have traveled, I owe to this lady, Professor Kamini Mendis, who took me under her wings from day one that I joined the department. And she has guided me. Even now, I reach out to her when I face challenges. So I am truly grateful to her. And there are other people who have helped my career enormously, like my co-supervisor of my PhD, Professor Richard Carter, Diane, so many from different parts of the world. I am really thankful for the confidence that they placed in me and the trust that they placed in me, giving me opportunities and so on. But I never could have done all this work on my own. And this is the team, colleagues from our department as well as outside, and the project staff. These are the more recent pictures, but there are so many in the past as well, and I remain very grateful to each and every one of them for their assistance with the work, including the international collaborators from various parts of the world, too many to name, and the college council members. I am truly grateful for their support in each and every initiative that we put forward, and I hope that there will be similar support that will continue for the rest of the year as well. I should specifically mention the role of the co-secretaries of the council, Dr. Nilakshi and Dr. Madhumani, who have worked tirelessly, particularly during the last several weeks coming up to this event. So I would like to thank them individually as well. Then the office secretaries, particularly Priyanka, who is a rock that we all lean on. She has very bravely shouldered responsibility almost single-handedly, because the second one, the junior person, left a few weeks back. So thank you, Priyanka, for everything. And last but not least, I would like to thank you for being here, sacrificing your long weekend. Thank you, and also thanks for your patient listening. Thank you.